Okay, so we're on page 86.
So the phospholipid bilayer creates the membrane?
Yes, and we're looking at membranes here. This is chapter 5. And the membrane is made up of millions of these special molecules. They're called phospholipid molecules. And phospholipid molecules are basically fat molecules. And I want to show you up close what those look like. You remember when we studied fats? You remember the parts of a fat? How many remember the parts of a fat?
Glycerol.
The glycerol. And Sierra, you had your hand up?
Fatty tails.
And the fatty acid tails. We take a glycerol and we put 3 fatty acid tails on it and we've got a fat, right? And if they're straight chained, what do we call them?
Saturated.
Saturated. But if they got a double bond in them, it makes them crooked. And what's that called?
Unsaturated.
So let's take an unsaturated fat here, and just for fun, let's pull off one of the fatty acid tails. And let's put on a nitrogen group, bloop, and a phosphate group, bloop. And you know what we have now?
Phospholipid.
A phospholipid. This molecule is one of these.
Does that have to be unsaturated?
It has to be one unsaturated and one saturated. I'm sorry, one saturated and one unsaturated.
So
So the whole thing's an unsaturated.
So it's a glycerol saturated and an unsaturated fatty acid, a nitrogen group, and a phosphate group?
You're right.
Okay.
Now I got a question.
Yes.
Why are the phosphate heads the ones on the outside?
What a great question. I was just going to talk about that.
Polar.
Here's the deal. If you look at these 2 groups on the top, the phosphate and this nitrogen group are both charged. The nitrogen group has a little positive charge, and the phosphate group has a little negative charge. And anything that's charged, we call it polar. So the top portion here is a polar, what we say is the polar head. This is like the head portion. It has a polar head, and it has these tails, these fatty acid tails, they aren't charged. So they're non-polar. And anything that's polar is going to be attracted to water, because water is a polar molecule, if you remember. Water is charged, and so anything else that's charged is going to be attracted to water. The negative part is going to be attracted to the positive oxygen, to the positive hydrogens, and the positive part is going to be attracted to the negative oxygens. So this part is attracted to water. And so we say it's hydrophilic. Hydrophilic means water-loving. Philia means to love in Latin or Greek or something like that. So this is a polar hydrophilic head, and this is a non-polar tail. It's not attracted to water, doesn't care for water.
Hydrophobic.
So it's hydrophobic. So hydrophilic is water-loving, hydrophobic is water-hating. So if you take a bunch of these phospholipid molecules and you just throw them into some water, they're going to naturally configure themselves into this arrangement here, with the hydrophilic heads facing outward and the hydrophobic tails all huddled together on the inside. There's water out here, there's water in here, and the heads all point toward the water and the tails point away from the water. They're kind of clustered in the middle there. These molecules will naturally configure themselves like this, on their own, without you having to do anything to them. If you just throw a bunch of fat molecules in the water, they'll configure themselves like this and form bubbles. Because you can imagine this will stretch all the way around and come together at the bottom, and you have a bubble. Fats naturally will form bubbles. You've seen this at the beach. You ever walk around and the waves are splashing? This is how I walk around at the beach. I'm pretty cool. Kick some sand in some little guy's face. And then I look and I see the waves crashing down and I see all that froth. You ever see all that froth? What shape is the froth?
Bubble.
Bubbles. Those are fat molecules that are lighter than the water and have floated up to the top from dead plants and animals and stuff in the ocean that have made them and released them, and they float to the top, and then they come crashing and they form these bubbles.
Like the foam.
The foam. Sea foam.
So it's from dead
Foam is like
things.
Fats. Yeah.
From dead things.
Yeah.
No.
Plants and animals and such. They make these organic molecules and release them. There's some other stuff in there too. Isn't that cool? Huh. So that's the basic structure of the membrane. And so that's what's around every cell, and that's what makes up all the internal membranes of the cell as well. It's all made of the same stuff, phospholipid bilayer. Now that's not the only thing in the membrane. You also have proteins scattered all through the membrane. And these proteins are, do you know what a protein is? Do you remember from the last essay? It's a chain of amino acids. All folded up. Let's fold this baby up. And stuck right there in the membrane. And some of them have a certain shape with a little hole in the middle, kind of like a hot dog bun, with a little hole, open space in the middle, and things can get through the membrane by going through that hole. They act as little doorways and such in the membrane to let things through.
Well, what do they do with things that aren't allowed to go through that try to get through?
Well, if it's a big molecule that can't squeeze through here, and it's not able to penetrate this part of the membrane, it's not going to be able to get in. Now, the cell does have ways of taking in large molecules. It can do what's called an endocytosis. It can kind of pinch inward and surround it in membrane and pull it in.
What keeps the proteins there?
The proteins are often anchored there. There's bits of cytoskeleton down here that can attach to a protein and anchor it in the right place. And also attached to a lot of the proteins, and just sometimes just attached to a phospholipid, are these little carbohydrate chains. Do you remember carbohydrates? We already studied them as well. Remember these hexagons all attached together in a line? Jessica, you hanging with me here? Good teaching going on. A chain of carbohydrates will sit here attached to every cell. And what those are, they're identification for the cell. They're identifiers. There are cells like white blood cells that crawl around and feel the cells. And they feel these chains. And if the white blood cells recognize the chain, they leave the cell alone. But if the white blood cell feels a carbohydrate chain it's never seen before, it'll think that this is a foreign cell and it'll kill it. If you saw that video I showed at the beginning, the white blood cell kind of crawling around in the body, it's feeling the cells, you see. And if it feels a cell with foreign chains, it'll kill it.
So those chains are basically like IDs.
They're ID, they're identifiers.
So
if I took Mac's kidneys, because I have bad kidneys, and so I'd kill him and I'd grab his kidneys and I'd get them installed and implanted by some black market surgeon that I hired. Well, it's probably not going to work because his cells don't have the same identifiers mine do. And my body will reject it. Have you ever heard of a rejected transplant? So people are sitting there waiting around on lists to get a matching donor that has the same identifiers as the person who needs it. Usually the matching donor will be someone in your family that has the same genes as you, so will likely have the same identifiers. That's what all that's about. It all has to do with these carbohydrate chains. And usually you find the carbohydrate chain sticking outside the cell, not inside, because it's outside where you need to identify yourself. Yes?
So is that why in an organ transplant you need immunosuppressant drugs? Or is that what they
That's correct. They give you immunosuppressant drugs to keep your white blood cells from attacking.
Yeah.
In some people, if you get a donor that's matching, but only sort of matching, not quite matching, you have to take immunosuppressant drugs the rest of your life, which means you get sick from other stuff pretty easily. But it's better than dying from not having kidneys. Yeah?
What are the carbohydrates attached to?
Some of them attach to proteins. Here you see one attached to a protein. And some of them just attach to the membrane themselves. They'll attach to special fats, and they'll just be hanging right off the fat.
Now, what kind of protein, or what kind of carbohydrates are they important to test?
Just
Just
like
random.
this. Carbohydrate chain. They probably all have specific names, but I don't know them. That's higher level college classes. You get into real cell biology, they get into exactly that.
What's the point of the carbohydrate chain and a glycoprotein if they're like kind of the same but not proteins?
If the carbohydrate chain is sticking off a protein, it's called a glycoprotein. If the carbohydrate chain is sticking off a fat, it's called a glycolipid.
So you just add glyco.
Glyco means carbohydrate chain. Let's see a few videos on this. Or not. Okay, that one's not going to do it. That was lame. I'm not seeing that one.
The machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
Wait, so the proteins are constantly moving?
Yes, all of these are constantly moving, or can constantly move. Some of them are stationary if they're stuck to the cytoplasm. But others aren't. They're just kind of floating around there. So they call this whole model of the cell membrane, it's called the fluid mosaic model. Fluid means moving. All these molecules are moving. And mosaic, meaning it's got a bunch of different pieces in it, just like a mosaic that you do in art. It's got some protein, it's got some carbohydrate, it's got phospholipid. It's the fluid mosaic model. So these, it showed good in that video I showed you at the beginning of class. It showed those molecules kind of moving around. Imagine if we got 100 people here in the classroom and we told them all to close your eyes and just kind of move around like this. This is, by the way, this is how I dance when I'm at Ziggy's. But anyway, you're just kind of bumping into things, or at a rave, when I go to one of y'all's raves that y'all have. So if there are a whole bunch of people doing that at once, that's what the membrane kind of looks like. It's all moving. And you saw that big, I don't know, it was like some kind of metal thing that was bashing the cell, and the cell would come apart. But then it'd come back together right on its own. Because because of the polar and nonpolar nature, it'll just come right back together on its own. There's another molecule in the membrane that you need to discuss if you're talking about the membrane. It's called cholesterol. And cholesterol you've probably heard of before. This is what it looks like. It's got kind of a weird look to it. It's actually based on a fat. It's a type of lipid molecule. it has a design, a steroid, looks just like this too. But it sits in the membrane and stabilizes the membrane.
How?
It keeps the membrane from getting too... remember, if you heat something up, the molecules move even faster? And so if these molecules start moving too fast, the membrane will start to fly apart. But the cholesterol makes some attachments here that keep the thing from flying apart. Also, if you get the membrane too cold, these things will stop moving and the membrane will get stiff. Well, then the cholesterol changes the way it bonds or something. I'm actually not exactly sure how it does it, but it keeps it more fluid when it gets cold. So it stabilizes the membrane when it gets either too hot or too cold. The cholesterol keeps it from getting too stiff or too loose.
So when things get too hot, the membrane falls apart?
Yeah, the molecules start moving
Is
faster.
that how you can get, like, you overheat?
Yeah, that's bad for you. You can't get it too hot. Your membranes will fly apart.
That's how you die from it?
Well, there's a lot of ways. Usually it's from proteins denaturing. Proteins coming apart, special enzymes that do things in the cell.
So why is too much cholesterol bad for you?
Well, what will happen is once your cell membranes have plenty of cholesterol, the rest of the cholesterol just float around inside your blood and form hard deposits that clog up your vessels. Okay. And that's not good for you. So what our problem is in America, and anyone who eats a lot, you have too much cholesterol, more than you need.
What's the difference between an integral and a peripheral protein?
An integral protein goes all the way through the membrane. A peripheral protein just kind of sits on top or on bottom. Whoops. That shows kind of what cholesterol looks like compared to the rest. There's a protein going all the way through the membrane there. A transmembrane protein, or an integral, that would be an integral protein. It shows on the bottom of 87, figure 5.2, it shows an experiment that was done to prove that the proteins could move around. This is kind of an interesting experiment. They took a mouse cell and they radioactively labeled the proteins of the mouse cell. And they show that by making them purple here. And then they took a human cell and they radioactively labeled the proteins in the human cell. And then they let the two cells fuse together. They let them combine in a petri dish. And cells will do this if they're put together in a petri dish. And so now you have this new cell that's both mouse and human cell combined. Now that's not going to grow into some weird mouse human that's running around eating cheese. That would be cool if it did. But one thing it will do is it'll show if the proteins are stuck in one spot, if they can't move, then this cell, the left half will be purple and the right half will be red. But the proteins can move. So after a while, they all mix together, and you have a mix of red and purple proteins all over. That proves that the proteins can move. Isn't that a cool experiment?
We should do it.
That would be fun to do. I don't know how to pull that off. You have the right equipment. On the bottom of page 88, it shows 6 different types of proteins that you would find in the membrane. If you were writing me an essay on this stuff, I would want you, and I said on the essay, if I said, talk about all the different membrane proteins in a page. Well, here they are. A channel protein is a protein that forms a hole where stuff can go in and out of the hole. allows a particular molecule or ion to cross the plasma membrane freely. Have you ever heard of cystic fibrosis? It's a disease. An inherited disorder is caused by a faulty chloride channel, a thick mucus collection in airways and pancreatic and liver ducts. Cystic fibrosis, you'd know it if you had it. It's inherited at a very young age, and people with cystic fibrosis usually don't live very long. Some people do, but most die when they get about teenagers. It's a terrible disease. What is this video? I don't know what this video is. Oh, this video shows stuff going through the membrane.
There are 3 types of passive transport through biological membranes. In one type of simple diffusion, lipid-soluble molecules pass directly through the phospholipid bilayer.
Now, some molecules can go through the lipid bilayer. Not a lot of them. They have to be fat-soluble, which means non-polar molecules. Can usually go through the membrane if they're small enough.
In another, lipid-insoluble ions pass through channels, proteins with an aqueous passage. In facilitated diffusion, a protein carrier moves solutes through the membrane by undergoing a conformational change.
How cool was that? It's fun to play with. See it going through? Sometimes the proteins will change their shape to let things through. But sometimes it's just a channel. It's just an open spot that anything can go through. This kind of protein right here is called a carrier protein that actually carries the thing through. It identifies it, and it'll only let it through if it's the right shape. I think that's one of our next proteins. Yeah, carrier protein. Selectively interacts with a specific molecule or ion so that it can cross the plasma membrane. The inability of some persons to use energy for sodium-potassium transport has been suggested as the cause of their obesity. Some people might be overweight because they don't have the carrier proteins aren't working right. I got another video showing you carrier protein working.
There are 2 main types of transport proteins, channel proteins and carrier proteins. Channel proteins are tube-like and provide openings through which small, dissolved particles, especially ions, can diffuse. In general, each type of carrier protein has a shape that fits a specific molecule or ion.
Why did it stop? What in the wide, wide world of sports is going on? It's not responding. Here's a cell recognition protein. A protein with a carbohydrate chain on the top of it. Already talked about those. Organ transplants are difficult to achieve if you don't have matching proteins. Here's a receptor protein. This is a protein with a certain shape that allows capture of some molecule in the environment. You see, this molecule is maybe a hormone, maybe like growth hormone. And it has to attach to this protein. And when it attaches to this protein, it causes the cell to multiply. If you aren't excreting enough growth hormone, your cells won't capture it and won't multiply. Or, if this protein's shaped incorrectly, you won't capture it, your cells won't multiply, and you'll be stuck real short, like a midget or a dwarf. They have a problem with their receptor proteins. Pygmies are short, not because they do not produce enough growth hormone, but because their plasma membrane growth hormone receptors are faulty and cannot interact with growth hormone.
What exactly is a pygmy?
A real small person.
I was just wondering, because my mom says that my grandma locks herself in her house because she's afraid pygmies will get her.
She's afraid pygmies will get her? She might have something wrong with her hormones. What's that?
Do you have contact solution?
I do not.
Contact
I'll have some in my
sorry.
book bag if you want.
As long as
Can you keep one eye closed and look with the other?
Put it in your mouth. Put it in your mouth. I've done that.
Do you want to go take it now?
I mean, I'm fine.
Okay. Here's an enzyme protein. And you see an enzymatic protein does a chemical reaction. Something comes in and is changed by the enzymatic protein. Enzymatic proteins will change things. And that's a chemical reaction. The little hat comes in and changes to a triangle, you see. Cholera bacteria release a toxin that interferes with the proper functioning of adenylate cyclase. Sodium ions in water leave intestinal cells and the individual may die from severe diarrhea. Gosh, that sounds unpleasant. Oh, I don't have a picture of junction proteins. It's on the bottom right of page 88. Y'all see those?
Junction proteins?
Junction proteins. It's where one protein and another protein kind of come together.
From two different cells?
From two different cells. So that stuff can be passed from one cell to the other. Y'all see that picture on the bottom right of 88?
What does the junction protein do again?
It's where two proteins are kind of lined up with one another so that stuff can pass from one cell into the other. Now, this next part talks about what can get in and out of the plasma membrane. And you can see here that charged molecules and ions they're not able to go through the membrane. If it has a charge, it will not be unable to go through. These fatty acid tails, being nonpolar, are not going to be attracted to those, and they're just not able to get through. Water is a semi-charged molecule and can go through, but it goes through really slowly. Water can slowly go through the membrane. Now, there are proteins that just let water right in. They're called aquaporins. And I think we talk about them later, but water can go right through the proteins pretty easily. If it's a non-charged molecule that's nonpolar, it'll go right through the membrane. Gases, oxygen gas and carbon dioxide gas will go right through the membrane. If it's a big molecule, what does that look like to y'all?
Macromolecule.
It says macromolecule, but what macromolecule does it look like?
Bacteria.
That's a protein.
I'm just kidding.
Quaternary.
That is. It's a tertiary structure. It may be a quaternary structure. But it can't get through the membrane. So large molecules don't go through.
What's the point of peripheral proteins if they just sit there?
No, often they connect to things or excrete things, or they're usually doing something.
Peripheral proteins, you said?
Yeah,
Where are those found?
on the outside of the membrane or on the inside of the membrane.
Where in the book
Kind of
is
sticking
that?
off of the membrane.
Where in the book?
Page 86.
I don't know. 86? On the bottom of page 91, it shows somebody dropping a crystal dye cube into a fishbowl. And the dye starts spreading out in the water, and eventually the whole fishbowl turns purple. I don't know why they would do such a thing to their fish. Isn't that kind of a funny picture? Bottom of 91?
They could just do it without fish.
Yeah, but they went ahead and did it with a fish in
Gonna
it.
kill a fish?
Probably not. Anyway, whenever molecules spread out, we have a name for that. It's called diffusion. Diffusion is molecules spreading out, moving from an area of high concentration where the cube is to an area of low concentration in the solution. Molecules spreading out. We call that diffusion. I've got some videos of diffusion happening. a terrible video. It's not much.
That was pretty bad too.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
So, if it was hotter, what would that do to the speed of diffusion, do you think? Speed it up, because the molecules move faster the hotter it is. All molecules are constantly moving. So raise the temperature, and you're going to raise the speed of diffusion. Diffusion is a very important process. This is how things get from one place to another. If you take a lump of sugar and put it in your stomach, it will diffuse out and spread all through your stomach. If you breathe in oxygen into your lungs, it will diffuse out of your lungs and into your bloodstream just by spreading out. It spreads out into your bloodstream. And so the oxygen gets into your blood that way. So it's a pretty important process. Here it shows you air coming in. This is top left of 92. Spreading out into the little lung pockets and then into the blood. And the blood, see, the blood vessels are surrounding the little air pockets in the lungs. So the oxygen spreads and gets into the blood, and the blood takes the oxygen to every other cell in your body that needs it. That's how the lungs work. Quite interesting. We'll study the lungs a little bit more later. So, we're running out of time. I guess there's the bell. So just read 5.1 and 5.2. I didn't get quite through 5.2, but we'll keep going tomorrow. Okay? Thank you, Don.